Presented by Caltech.
My name is Steve Mayo, and I'm the chair of the Division of Biology and Biological Engineering at Caltech. And I'd like to start tonight's Ernest C. Watson lecture by thanking all of you for taking time to attend tonight's lecture, which will be delivered by Pamela Bjorkman, the Max Delbruck Professor of Biology at Caltech and an investigator with the Howard Hughes Medical Institute. The Division of Biology, the predecessor to the current division, had its origins in the late 1920s and was founded on the idea of using basic research to elucidate the fundamental principles of life. Over the decades, faculty in the division have excelled, winning major awards for work ranging from discovering the relationship between our genes and the function of cells in our bodies, to how viruses work, to how genes control behavior, and even the discovery of the basic idea that different parts of our brains are specialized for different functions. To date, faculty in the division have won 6 Nobel Prizes. Even though the focus of the division has been on fundamental discovery, the division has always been mindful of the potential impact on society. 2 important historical examples are the creation of the field of molecular biology, which is the foundation of the biotech industry, and the invention of the automated DNA sequencer, which was used to sequence the human genome and is the underlying technology in the developing area of personalized medicine. 2 years ago, the division changed its name to the Division of Biology and Biological Engineering. There were many reasons for this change, but the one that is most relevant for tonight is that the new organization recognizes and therefore enhances the ability of faculty in the division to move seamlessly from research focused on basic discovery to engineering or research that is focused on leveraging fundamental insights in the service of society. Pamela's work is a great example of this new paradigm. Her early work focused on primarily understanding the molecular details of how our immune system works, and she's made major discoveries in this realm. Pamela is now leveraging these discoveries through biological engineering in an attempt to benefit society. We'll hear a lot more about this in a moment. Pamela received an undergraduate degree in chemistry from the University of Oregon, a PhD in biochemistry and molecular biology from Harvard University, and did postdoctoral work both at Harvard and in the Department of Microbiology and Immunology at Stanford University School of Medicine before starting at Caltech in 1989. She has received numerous awards in recognition of her accomplishments, including the 1994 Gardner Foundation International Award for Achievements in Medical Science. She was elected to the National Academy of Sciences in 2001, which is a widely accepted mark of excellence in science and is considered one of the highest honors that a scientist can receive. The title of Pamela's lecture tonight is A Molecular Arms Race, the Immune System versus HIV. Without further ado, please welcome Professor Pamela Bjorkman.
I want to thank everyone for coming tonight. And what I'd like to do is tell you how we're thinking about the war, which it really is a war between HIV and our immune system. And I just want to start by reminding everyone that although it may not seem like it in the United States, HIV/AIDS is still a worldwide problem and it needs to be addressed. So there's actually over 35 million people today who are infected with HIV, which is the virus that causes AIDS, but only 37% of them or so, just a little over a third, are being treated with the available medicines, which are anti-retroviral drugs. And you can see that this is a huge problem, especially in sub-Saharan Africa. So to explain why this is a problem and why our immune system has such a problem fighting this virus, I want to introduce you to the villain in this story, which is the HIV virus itself. So HIV is a so-called enveloped virus, and that means that it stores its genetic material inside a lipid membrane, and that membrane is derived from the host cell. And the genetic material in the case of HIV is ribonucleic acid, or RNA, and it's inside the virus right here. And then the lipid membrane that I referred to is this area right here, which encompasses the virus and protects the genetic material. This is what I'm going to be talking about a lot tonight, which is the so-called envelope trimer. So the virus is called an envelope virus because it has this membrane. And then this trimer right here, I'll either call it an envelope trimer or a spike trimer. And it's got two subunits that I'll be referring to as GP120 and GP41. So the purple one is involved in docking to the host cell that it's going to infect, and then the green one transverses this lipid bilayer and then is involved in fusing the virus to the host cell and then getting the genetic material inside. So I want to describe the HIV life cycle a little bit because I think it will illustrate just how dangerous this virus really is. So you start off and you get an infection from what is a free virus. And then that attaches to a host cell. And the way it attaches is this one of the protein components of the spike trimer, the thing I called GP120, that binds to a receptor on a host target cell, which is called CD4. And then it goes inside, makes copies of its genetic material, makes more copies of virus, and comes out. I'll go into that a little bit more. But what I want to talk about now is why it's so dangerous that the receptor for this virus is CD4. The reason it's so dangerous is that the use of CD4 as the receptor for HIV targets this virus to cells of the immune system. So HIV infects so-called CD4 positive T cells. T cells are a type of T lymphocyte. They're a lymphocyte or a white blood cell. But the problem is that they control other immune cells. And so if you start to wipe out your T cells, you will eventually get just complete immune dysregulation. So the typical course of an HIV infection, you would start out and a healthy person has maybe 1,000 CD4 T cells per microliter. And upon initial infection, that goes down a lot, and the person may feel like they have the flu or something like that. But the T cell count goes back up again because the immune system sort of recovers. But then over the course of many years, it just keeps going down until when it gets below about 500 T cells per microliter, they may have symptoms, and then below about 200, that results in AIDS, which is acquired immunodeficiency syndrome. So if this infection is not treated, what happens with this gradual loss of CD4 T cells is that you just lose your ability to mount what are called cell-mediated immune responses, and then you get infected with other things. So what kills the HIV-infected patient is not the virus itself, it's these opportunistic infections. And so HIV patients present with things like yeast infections, they get tuberculosis, they activate latent viruses such as the chickenpox virus, and then they get shingles. There's all kinds of things that happen to create B-cell tumors and another type of cancer through infection with a virus, other types of yeast infections leading to pneumonia, and so on. So this is just untreated. This is a terrible problem, and people eventually die from this. Now, the next thing that HIV has kind of a trick up its sleeve is that it's a retrovirus. And what retro means is that this virus reverses the typical flow of genetic information. So you may have heard of the so-called central dogma that Francis Crick described, which is DNA is transcribed into RNA and RNA is translated into protein. So the flow of genetic information is usually thought to be DNA to RNA to protein. But these retroviruses actually come in as RNA viruses, and then they do something called reverse transcribe their RNA into DNA. So this is the opposite of what people were expecting to find in any type of virus. It was thought in the 60s that it was impossible to reverse transcribe RNA, and in fact, our former Caltech president, David Baltimore, got the Nobel Prize for discovering this enzyme that reverse transcribes RNA into DNA. Okay, so because it reverse transcribes the RNA into DNA, the problem with this enzyme is that it's very error prone, so it makes a huge number of mistakes, which are what's called mutations. So This rapid mutation rate is actually amazingly advantageous for this virus. It gives it a huge survival advantage. And so in general, one of the things I want to emphasize in this talk is evolution, in fact. So HIV is an example of a marvelously evolved little creature. The reason that is because genetic changes in general are what drive evolution. So it allows, in this case, HIV to adapt to changing conditions, and what that means is what your immune system is throwing at it. So from the perspective of one of us, so we don't have very many progeny and we don't reproduce that fast. Mutations aren't that great, right? Because mutations, if you make a permanent genetic change, that's a mutation. That can result in things like sickle cell anemia. This is a single nucleotide change in DNA that changes one amino acid in the oxygen-carrying protein hemoglobin in your blood. Profound consequences. And then cancers are caused by gradually accumulating random mutations in DNA. So that's us. But from the perspective of the virus, this is really great, because the virus is going to make millions of progeny, and it doesn't care if 99.99% of these are unfit or they're killed by your immune system. As long as one or some small percentage can go on, that allows emergence of really rare variants that are more fit than the parent, or they can evade the host immune system more efficiently. And it's because HIV has, its reverse transcriptase has this enormous error rate that it's such a problem. One of the reasons it's such a problem for our immune systems to deal with. Now, another thing that HIV does is what all retroviruses do. It takes the DNA copy of the RNA and it integrates it into the host genome. So what does that mean? Well, the cells are making new viruses, and these viruses are coming out. They're coming out as many different variants. But this cell, even when it's done producing viruses, it has a copy of HIV DNA in its genome. So every single descendant of this cell carries HIV DNA. And so if you take anti-retroviral drugs, you will gradually get rid of all the free virus in your body. It goes to undetectable levels. But you can't get rid of this integrated DNA. And so as soon as the person stops taking the anti-retroviral drugs, eventually that's activated to produce more viruses. So you cannot stop taking the anti-retroviral drugs, and it's because of these latent reservoirs here. Now, fortunately, as I've alluded to, there are drugs that people take. These are called, as a collection, they're called highly active antiretroviral therapy, or HAART. This is given in combination, usually 3 or more different drugs. And the drugs are inhibitors of this enzyme reverse transcriptase, or they're inhibitors of the integrase. They're inhibitors of a protease, which is something that cleaves the HIV proteins. They can be inhibitors of actually binding and fusing to the host cell membrane, and so on. So a person would take a combination of these, and the reason is that if they take just one, the virus mutates against that drug and then survives. But if they take 3, the virus can't mutate fast enough to overcome 3 or more at once. Here's the problem for today's world. Only a little over 1/3 of infected people are being treated with these drugs. So we need something different. And ideally, what we need is a vaccine. This would prevent people from getting HIV in the first place. And so the way any vaccine works is you inject something into the person. It could be an inactivated virus, or it could be a recombinant protein subunit or something like this. But then your immune system inside your body makes antibodies, and those get rid of the virus eventually. So that's the way, say, flu virus vaccines work, or poliovirus vaccines work, and so on. And if that worked in the case of HIV, and you made these antibodies, they would interfere with attachment of the virus to the host cell in the first place, and so you would prevent the initial infection. So I want to tell you a little bit more about antibodies, because that will be a major focus of the rest of the talk. So antibodies are proteins, one form of them are proteins that are in your blood, and you produce a lot of different kinds of antibodies. And the antibodies evolve with your immune system to be specific to whatever pathogen that you encounter today or the ones you will encounter next week. And there's an amazing mechanism to guarantee this. If you have an antibody that's specific for a particular virus, it will do something called neutralize that virus. And that means that it prevents the virus from infecting the target cell and from making new viruses that would go on and infect other cells or infect another person. And so in order to illustrate the role of antibodies in the adaptive immune system, what I want to show you is that an antibody can be made against anything that you will ever encounter in your lifetime. And this was very graphically illustrated in the movie Fantastic Voyage, if any of you saw this movie. In this, what you're seeing here is a clip in which antibodies are attacking Raquel Welch. She was miniaturized and put into a miniaturized submarine that was injected into a scientist who needed a blood clot removed, and so all these other scientists came in with her, and she went out of the ship once to do something, and these antibodies there attacked her. So clearly, if the antibodies can be specific to attack Raquel Welch, then they could attack anything. Okay, so it's really amazing that we as mammals have the capacity to make, at least 10 to the 16th different types of antibodies against anything you're likely to encounter. And in the rest of the talk, I'm going to be showing you the differences in antibodies by coloring them differently. And then I just want to introduce several parts of the antibody that I'll be talking about. I'll be talking about the so-called Fab arms. So there are two variable, this part of the antibody varies, the Fabs, and the Fc remains relatively constant. And the Fabs are what bind antigen, and that can be illustrated by the name. It means fragment antigen binding. I won't talk so much about the Fc, but it has important roles in recruiting immune cells once an antibody is bound to a pathogen. Okay, so this is the part that varies from one antibody to another, and it's the part that would be specific for one virus versus another. Okay, so let me show you what happens when your immune system encounters HIV. You get infected with a virus, and inside the person who infected the other person, there are thousands or millions of different viruses because of this rapid mutation rate. But really interesting studies have shown that usually there's only one virus that gets across to start a new infection in any new individual, and that's called the transmitted founder virus. So in this case, that's this yellow thing right here. Okay, so it starts infecting host cells, and it reverse transcriptase makes errors, and it makes a bunch of different types of viruses right here. They're all kind of similar to each other, so colored them in various shades of yellow. So a couple of these things are not fit, so they just die. But remember what I said, the virus doesn't care about that. Other times, antibodies will get rid of these viruses here, and so those are gone. But all you need is just some minor fraction here to go on and survive and to make more viruses. And now some of these will be unfit, some of them will be attacked by antibodies and they'll go away, but others will produce more types of viruses. Some of those will be unfit or attacked by antibodies, but those will go on and produce more viruses, and this will keep going on until you have what's called a viral swarm inside any infected individuals. And so that's illustrated by all the different colors of viruses here, and then all these different colors of antibodies. But the problem with these antibodies is that most of them are strain specific. So this cyan-colored antibody might kill this virus, but it won't touch these here because it doesn't even recognize them. And so this amazing diversity of viruses inside a person is just an astounding thing for both the vaccinologist to deal with and also for the person to deal with. And so just to give you an example, there are more strains of HIV in a single infected person than there are influenza strains in the entire world. And so can you imagine trying to make a vaccine against something that is that diverse? It's really a daunting problem. Okay, so I've mentioned that the HIV-infected people make all these different antibodies, and we'll just look at the antibodies now. These have been characterized by a lot of different labs across the country and in the world. They find out their molecular details through the wonders of DNA sequencing, which is a way to get the sequence of the antibody protein. And so once they characterize these, they ask, how well do they work? And most of the antibodies turn out to be specific for an individual or a couple of strains of HIV, but some of them, from rare patients, are broadly neutralizing. So let's say this one is a broadly neutralizing antibody, and that means it neutralizes, say, 60 or more % of HIV strains instead of, say, 2 to 5%. We call these bNAbs for broadly neutralizing antibodies, and I'll be referring to these throughout the talk. Now, broadly neutralizing antibodies, as I said, they're produced by a rare population. If you take HIV-infected individuals, it's estimated that maybe 5 to 10% of them, and that's all, will ever develop these types of antibodies. And it takes them 2 to 3 years of infection to develop them. But if you take those purified antibodies and you put them into a macaque model, this is a non-human primate, a macaque model of HIV infection, they will actually protect these primates in a virus challenge experiments. So if we could make a vaccine that raised these broadly neutralizing antibodies, that would be the end of HIV in our world. And we can't yet make a vaccine that will elicit these broadly neutralizing antibodies in either animal models or in humans. And it's a daunting problem. People are still working on it, but I'm going to talk about another possible way to get around that problem. And so the way we're trying to get around this problem comes back to fundamental research in biology. And the way we think about it is the way Richard Feynman said, he said in 1959 actually, about biology in general. It is very easy to answer many of these fundamental biological questions. You just look at the thing. So what I'm going to tell you about is looking at HIV, which is a virus, and looking at antibodies, which are proteins. And I'll have scale bars on most of the slides because we think in terms of distances, and these are very small distances. This is 100 to 150 nanometers, and nanometers 10 to the -9th of a meter, so it's quite small. And then the antibody is 10 times smaller than the virus. And so we look at these things in a variety of different ways, but what I'm going to tell you about is one way that we can look at proteins from the virus and also the antibodies themselves using a technique called X-ray crystallography. This is kind of a form of microscopy. It's an experimental technique where you can determine a three-dimensional structure of a protein or a nucleic acid or a complex between the two or a complex between two proteins or whatever. You can do this at so-called atomic resolution. And what that means is that through growing crystals of whatever your protein is, getting a diffraction pattern, you can put this into a computer to calculate a Fourier transform and then get what's called electron density, which you build to locate where all the atoms are in your protein, and that might be 10,000 or more atoms. And then you look at the three-dimensional shape of the protein, and this has a variety of helices. These are called alpha helices, a common secondary structural element in proteins that was discovered by Linus Pauling when he was here at Caltech. Okay, so we can locate all the atoms. That happens to be a phenylalanine ring of an amino acid, and that will become important for a little bit about what I'm going to talk about later. But so what I'm trying to tell you is that we have the ability to look in 3 dimensions at what these proteins look like, and we can study the chemistry of how an antibody recognizes an HIV protein. And so over the years, we've been looking at HIV proteins and how they are recognized by antibodies, and the goal of this is to define what are the structural correlates of broad and potent neutralization of HIV. So we have antibodies that neutralize well. We have antibodies that neutralize sort of well. We have antibodies that are horrible. They don't neutralize very well at all. And so we solve three-dimensional structures of HIV proteins bound to these antibodies, and we try to figure out what's going on. And so these are I'm not going to discuss individual structures here in this talk, but over the years, these are people who've determined some of these structures, and I want to thank, of course, the great facilities at Caltech that make this possible, the Molecular Observatory, and also electron microscopy facility. And both of these are funded by the Gordon and Betty Moore Foundation and also by the Agaron Institute. And then we have to make all these proteins. These are done by a protein expression center that's in the Beckman Institute, just right around the corner from here. Okay, so why are we doing this? Because maybe we could use insights gained from studying the structures to design better broadly neutralizing antibodies. And I have to say that as a chemist, I really like looking at these interfaces and understanding the chemistry behind it. But I'm going to kind of focus on the practical applications here, but I'll come back to the basic biology at the end of this. So the idea is if we could make better broadly neutralizing antibodies, they're so rarely made by people, can we actually make them better? And then we could use them as weapons against HIV. So I told you that right now we can't make a vaccine that would elicit these broadly neutralizing antibodies, but if you administer them in an animal model, they protect. So why not administer broadly neutralizing antibodies to people? And if you're going to do that, you would want to administer the very best possible ones. So we're trying to improve them. But first I want to say that it's not completely outrageous to think that we could administer these purified proteins or do it through some sort of gene therapy method. It's not completely unreasonable to think that we could administer them to either prevent HIV infections in the first place or to treat an ongoing HIV infection. The reason for that is just that we've been doing this as human beings for over 100 years. The first use of antibodies to treat an infectious disease was in 1895. This was called diphtheria antitoxin. It's just serum antibodies, and these were raised in guinea pigs or horses or whatever, and they were given to people to treat diphtheria. Now there are a number of FDA-licensed antibodies that are approved for either treatment of an infectious disease or prevention of that infectious disease. Okay, so in thinking about why we want to engineer these antibodies, let me explain how we think about this, and this comes back to the title of the talk. So we think about this as a serious war between the human immune system and the virus. The human immune system is incredibly complicated. It has organs, spleen, thymus, and so on, lymph nodes, and I don't have time to talk about any of that. But it's extremely complicated and it's very highly evolved. This virus has gone through many more cycles of evolution than our immune cells ever have. So as I'm going to show you, right now it has the advantage over us. Okay, so if you look at what the immune system does, it has something called cell-mediated adaptive immunity, where a bunch of different types of cells work together to get rid of infections. And they do it specifically, and they have memory so that the next time you're infected with the same thing, you get rid of it more quickly. That's the basis of a vaccine. Okay, so what does this virus do? Well, the cells that it infects are T cells, and these are absolutely critical for immune responses. They regulate pretty much your whole cell-mediated immune system. So the advantage goes to the virus in this case. Okay, what else? Your immune system has lots of methods to clear viruses. Some of them have nothing to do with T cells. In fact, most of them have nothing to do with T cells. And so we ought to have the advantage here. However, remember I told you that HIV is a retrovirus, so it integrates its genome into your host cell. So that means that you have latent reservoirs of cells that have HIV genes in them that can be activated at any point. So this is an extremely difficult virus to get rid of. So advantage to the virus. Okay, now we have antibodies. And antibodies are wonderful things. They can neutralize viruses. They can recruit cells that bind to the antibody and then kill the virally infected cell. Maybe they should be able to get rid of latent reservoirs. But there's all these antibody evasion mechanisms that this virus uses, including rapid mutation that I talked about. And then the parts that have to stay the same for the virus to be the virus that it is, it hides them in places the antibody can't access. so the advantage goes to the virus. So we want to do something that the virus has not evolved to fight against. And what I think is that if we make unanticipated weapons, this virus, this virus has never seen before. I'm hoping that it can't evolve fast enough to get around what we're trying to do, which is to engineer these antibodies to protect against HIV, or to even treat an ongoing HIV infection. Okay, so to do this, so hopefully we have the advantage here. That's what I'm going to try to say. So to do this, I'm going to give you a short snapshot of one way to do this, and this is kind of basic chemistry, where what we're trying to do is take a look at the molecular structures of how these antibodies work to block the virus from infecting, and then see if we can improve them so that they do a better job. So what I'm going to show you is a movie of starting with the structure of the GP120 part of that envelope trimer spike that I've been talking about. This is the HIV protein, right here in gray. And right here is the host receptor, CD4. And so this is a crystal structure. This is derived from a X-ray diffraction experiment where we know all the three-dimensional, we know the three-dimensional shape of this. We know where all the atoms are. We're showing it in a sort of schematized way. And so when I run the movie, what you're going to see is that the receptor binds to GP120 in a specific way. And this particular class of antibodies that we've been working on is a mimic of the receptor, so it blocks the receptor from ever binding. So that's how these antibodies work. So these are called CD4 binding site bNAbs. So the way they work is that if they bind to the GP120 can never bind to the CD4, and so you never get an infection, okay? So I'll start the movie, and what it's going to show you is that there's a crevice in the GP120, and the CD4 puts a phenylalanine residue. Remember, I pointed that out. This is a hydrophobic residue. It's basically a benzene ring, and it sits in this hydrophobic pocket, meaning it likes to exclude water. The antibody had nothing at this position, so it wasn't as good as the CD4. So what we did was to put a tryptophan into that position. A tryptophan is also a hydrophobic amino acid that has some properties in common with phenylalanine. And so it made, then the antibody looks like this when it's bound to the GP120. It filled that pocket there, which it used to have nothing in that pocket. So the antibody wasn't quite as good as it could have been. And then we tested it. And so I'm going to be showing you curves like this, and it's not as bad as it might look. What these are called are coverage curves. And so let me explain what we've done here. So what's done in each color is these are the results from neutralization assays against many different HIV strains. And so what I mean is we take an antibody and we assay how well it neutralizes a certain HIV strain. And then we take that antibody and we assay it against 100 more HIV strains. And then what we do is we take those results and we plot the percent of HIV strains that are neutralized here, percent coverage, as a function of concentration of the antibody that achieved the neutralization. So the best antibodies neutralize, well, the best antibody possible would neutralize 100% of the viruses tested. And these are a subset of viruses that circulate in people. And it would do it at very low concentrations because that means it's more potent. And so if none of that makes sense to you, you want to be as far over to the left as you can be, and you want to be as far up to the top as you can be. And that means you're the best antibody. And so these are, the arrows are pointing to 3 antibodies that we've engineered, and you can see they're over to the left. And they're pretty high up in terms of absolute coverage. And they started from this antibody right here, which is the one in dark green. And so you can see that antibody was pretty good to begin with, but we made it a lot better. And we ended up making these antibodies, these 3 antibodies. It's a first generation with the tryptophan, and then we made 2 second generation versions of it. And these really are better than any broadly neutralizing antibodies that were isolated from patients. So they're the best antibodies out there. These antibodies here or their very close relatives are being used in clinical trials now. And so these are actually more potent than them. So they could be used potentially at lower concentrations, and you'd have fewer side effects, and it might be practical to administer lower concentrations. You know, there's a financial aspect of this in terms of manufacturing the amount of antibody that you need. Okay, so we can do this structure-based design. That's great. But, you know, the fact is, there isn't a single broadly neutralizing antibody that achieves 100% breadth. None of them go up to here. And in experiments, it's very clear that HIV will mutate to become resistant to any single broadly neutralizing antibody. This is a horrible thing about HIV, is it will evolve so that we take our very best antibody and we throw it at a single strain of HIV. Eventually, mutants will come out in this category right there, and those will grow up and just be infectious. So we kept thinking about this. You know, we were pleased about making these better broadly neutralizing antibodies, and they might be useful in therapeutic settings. But we thought there must be something else going on here, because it's true that HIV has a lot of strategies for eluding antibodies, especially rapid mutation. And then the parts that aren't mutating are hidden at interfaces away from the antibodies, and it's got carbohydrates that antibodies generally don't really want to bind to. But we thought, you know, a lot of viruses are like that. Influenza virus is like that. It does all these tricks here as well. And so there must be something else going on. And by looking at HIV, we thought we understood what that might be. So that's what I want to show you. We think it has something to do with the low density of these envelope spikes right here. And so to understand what I'm talking about, I'm going to show you what other viruses look like. So typical viruses against which an antibody response can either clear an infection, or we can prevent the infection in the first place by raising antibodies through vaccination. They look like this, and you'll note that they have a lot of envelope spikes. They're presented as a dense array here, so lots of envelope spikes. Here's what HIV looks like. It's practically bald in terms of its number of spikes. It has on average 14. Flu virus is the same size. It's also a spherical virus, and it has on average 450. So there's something going on here, and we know that in general the antibody response against HIV is ineffective, and we know that the envelope spikes are few and, as we say, far between. So is this causally linked? Well, we think so, because antibodies actually bind best if they can use both of their arms. Remember, I said these were fragment antigen binding. These are the arms of the antibody, and this is, if you will, its body. And here's influenza virus. The way an antibody would bind to the neighboring spikes in influenza virus is it could reach between the spikes. And the analogy would be, if you were trying to hang on to something, you have two arms, and you'd do a lot better if you could hang on with both of your arms. So to really illustrate what's going on here, I'm going to play this movie of what happens to Elmo if he's hanging on to, if he can hang on with both of his hands to neighboring handles. So he may fall off every once in a while, but he's fine. Now the handles mutate, and he can't hang on as well, and so his hands are going to fall off more often, but he can still hang on because on average he'll always have one hand there. So that's the concept in biology of avidity. You have an antibody, it has two arms. If one falls off through a dissociation rate that is measurable, then the other one will probably be hanging on. Okay, so that's on a virus like influenza virus. Here's what we think would happen on a virus like HIV. So Elmo's fine as long as the spike handles fit his fingers. He may, you know, wave his arms every once in a while, but he's not going to fall off. But now, look what happens if the spike mutates or the handle mutates so that his hands don't fit very well. He's going to eventually fall off. So we put together an opinion piece which we called Few and Far Between, How HIV May Be Evading Antibody Avidity. And avidity just means the ability to bind to two different, to gain a high affinity through binding to two different things at once. And so the idea is that for most viruses, the fact that an antibody evolved to have two identical Fabs, or antigen binding fragments, that would permit bivalent binding through what we call inter-spike cross-linking between two spikes. But unlike other viruses, HIV has hardly any spikes, and the spikes that it does have are generally pretty far apart. So most antibodies can't bind with both of their Fabs. That's the hypothesis. So we think that HIV's low spike density plus its rapid mutation is a deadly combination. And so antibodies can initially bind, probably with just one arm, but they're eventually going to fall off before they can do any good. So the question is, maybe we could engineer an inter-spike cross-linking antibody. We could just, do something to lengthen the arms so it could reach between spikes. But this isn't going to work because, as you saw from these pictures, there's no constant distance between these spikes. So you can't make a single reagent that has the right length to reach between spikes. And so even on a single virus, that's true. And then when you get the thousands of viruses that there are, there's no way this is going to work. So instead, we want to make something called an intra-spike cross-linking reagent. This is something that would cross-link or bind twice within a single spike. And so if you could do that, it would look something like this. Now you have bivalent binding, which allows you to hang on tightly. And it doesn't matter that you have hardly any spikes, and it doesn't matter how far apart they are. So to illustrate this, we're going to go back to our analogy here. I switched now to a different type of handle. This is really the schematic of the spike trimer. And now there are two places that Elmo can hang on. These are called, in immunological speech, these are called epitopes. Those are where the antibodies bind. And so here Elmo, with his blue hands, is binding to the blue epitope. Here he's binding to the orange epitope with his orange hands. And here he's binding to one of each. And so the analogy I'm going to make in the reagents that I'm going to show you are, this would be called a homo-diFab, meaning if you think of these as the Fab arms of an antibody, they're the same here. It's binding to the same epitope on two different subunits. So I should re-emphasize here, this is a trimer, and there are three identical subunits here, so each epitope is repeated 3 times. We'd like to make a 3-handed Elmo, but that will be the topic for later. But right now we're making 2-handed ones. So the analogy here is a homo-diFab that binds to the blue epitope, or a homo-diFab that binds to the orange epitope, or this would be what we call a hetero-diFab. One binds to the orange and one binds to the blue. So now let's see how this would work if Elmo's trying to hang on. So he can't reach between the handles, but what he can do, if he's a hetero-diFab, is he can bind to 2 different epitopes, and he's not going to fall off. Well, he's not going to fall off because it hasn't mutated yet. Now it's mutated. His fingers don't fit very well, and so he's going to have a lot of this on and off for each individual hand, but it's not going to matter because he's able to bind bivalently, if you want to think about it in molecular terms. And so we think if we could make such a reagent, it would work, perhaps, even if HIV mutates. So let's see how we would do this. So how do we make an intra-spike cross-linking reagent that binds to a single HIV spike trimer? Well, we know something about the structure of these spike trimers. We know how large they are. We know the approximate distances between their epitopes, which is where the antibodies bind. But then if you look at, I'm only showing you this schematically, but I think you can see it even if I showed you the real 3-dimensional structure, it would be a bit more confusing. But I think you can really see it here. Here's a typical antibody. It's got these 2 arms. There's some flexibility between the arms, but there's just no way that a real antibody that your body makes is going to be able to bind to 2 adjacent subunits in this spike trimer. The architecture absolutely forbids this, which I think is deliberate on the part of the evolution of the HIV spike. So this isn't going to work with a normal antibody. But I think we can make engineered reagents that would bind in the way that I showed you. And so that's what I'm going to talk about. So first I want to talk about how we actually identify the distances between the epitopes, which are the antibody binding sites, on these spikes that are actually on the virus. And the reason we have to do this is that even though we have some ideas about the structure, it's actually known through the work of other labs and also in our lab, it's known what the structure looks like in molecular detail, but on the virus, it's breathing and moving. It's actually a dynamic protein. It has a closed state, it has an open state, it has other intermediate states. We don't know what's going on in the virus. Our pictures are static. They're from X-ray crystallographic or EM, electron microscopy images. Those are static. We don't know about the dynamics here. We need a ruler that will measure a distance between these two epitopes. And that would actually, from a basic research point of view, give us some information about envelope dynamics and conformational states of this protein, which we're very interested in understanding how it works for the virus. So if you were trying to measure something and you had your choice of a plastic ruler or a piece of yarn, which would you use? I would use the plastic ruler, and the reason is that the yarn can adopt different conformations. So we needed to find something that's on a very small scale here. We're talking about 10 nanometers or so, or even less in some cases. And so we needed something that was relatively rigid and would maintain its shape and would also have defined increments. And what really fits the bill is DNA, double-stranded DNA. This is the familiar, I hope, double helical structure of double-stranded DNA that you've probably seen in many textbooks. And so this, we would say, is the analog of a plastic ruler. It has 3.4 angstroms per base pair here. And so if we have 10 base pairs, we know that it's 34 angstroms, and so on. We thought about using proteins, and we tried, but actually it's about 3.8 angstroms per amino acid, but when proteins fold up, they fold into all kinds of different conformations where the distances aren't exactly known. And then in an unfolded protein, there's no way, it folds around like this. There's no way to use it as a ruler. So we started to make libraries of Fabs. These are the antigen binding parts of these antibodies, connected by different lengths of double-stranded DNA. And the DNA in this case is functioning to link these two together, but it's also functioning as a molecular ruler, and then the increments that it's measuring are these base pairs, which are 3.4 angstroms apart. Okay, so what you do is you take a Fab from an antibody, you link it to single-stranded DNA, which has an overhang, and then through the miracles of base pairing, which is completely and utterly understood, you add different lengths of double-stranded DNA with complementary overhangs. You mix these together. You get all different lengths now of these so-called difabs. And this is what they look like if you look at them under an atomic force microscope. This is what we thought they'd look like. They have a rigid piece in the center and then the Fabs hanging off. Then what we do is an in vitro neutralization assay with many different lengths of Fabs. So let me explain that for a moment. What you do is you have a target cell, and when it's infected by HIV, it lights up through a luciferase gene, actually. Okay, so now you add different concentrations of the potential inhibitor, which is an antibody, and the amount of luciferase goes down. So you have a measure of the degree of inhibition caused by that antibody. And so what you do is you plot the percent of the inhibition, which we're calling the percent neutralization, as a function of the concentration of the antibody you added. And you get a curve that you measure it at, maybe 10, 12 concentrations, maybe 8. You measure it here, you get a curve, and which one is the most potent in this example here? The green one barely neutralizes, it doesn't neutralize. The blue one's okay, but the red one is the best because it neutralizes at the lowest concentration. We characterize these by so-called IC50 values. IC50 means the concentration at which you get 50% inhibition, so you can see it's a lot lower for the red one than for the blue one. Okay, so now what we're going to do is an experiment where we compare the neutralization potencies of a homo-diFab as a function of the double-stranded DNA linker length. So what happens is you take the Fabs, you make all kinds of you make a lot of different ones with different lengths of double-stranded DNA connecting it. And at the right length, that is the correct distance between the epitopes, you should get this intra-spike crosslinking here. And so for each of these double-stranded DNA constructs, we then do an in vitro neutralization value assay, and we get the IC50 value. So then you plot the IC50 as a function of the bridge length, and you can see this really amazing minimum here, where this is where it's the most potent, and it's actually about 100 times more potent than the parental IgG, that's the red line here, from which it was derived. So there is an exact distance that's absolutely that would give you this, and the rest of these double-stranded DNA bridges are not quite the right distance, so in theory they're probably binding with only one arm hanging out there. And then just a few explanations here. This is one of these CD4 binding site antibodies. The Fab was derived from that, and I showed you that in the earlier movie. Okay, so the most potent neutralization seen was with a 62 base pair DNA linker. 62 base pairs times 3.4 angstroms per base pair is about 210 angstroms, and that corresponds to what's known from other people's work of the CD4 open version of the trimer on the virus. So we think this is what we're inhibiting. This was a surprise. We thought it might be that, but that suggests all kinds of things about how these antibodies neutralize that we'd like to pursue. So I just want to show you now a coverage curve you remember the coverage curve. If you don't remember, the further to the left it is, the better, and it should go up ideally to 100%. So this is the coverage curve for this homo-diFab, meaning the Fabs on both sides of it are the same. Sorry about the nomenclature here. It's a CD4 binding site antibody. That happens to be its name. Okay, so the 62 base pair double-stranded DNA linker was the best, and that's the coverage curve is this red one here. It's considerably over to the left than from the parental IgG. Both of these have two Fabs, but the difference is the Fabs in this construct are the right distance apart, we think, to achieve this intra-spike crosslinking. So now we're starting to make what we call Hetero-diFabs, which is what you saw in the Elmo movie. So the Hetero-diFab is something that's designed to bind to two different epitopes, and we don't necessarily know the right length of double-stranded DNA to connect it. So again, we made a bunch of different constructs with different lengths of double-stranded DNA. And one of them that worked quite well was linking this antibody, which binds to the top of the spike trimer, to this antibody, which is a CD4 binding site antibody that binds kind of to the side. And it was 50 base pairs that worked pretty well. And now this is a coverage curve for that Hetero-diFab compared to the two parental antibodies from which it was derived. So it has a Fab from 3BNC60. That's just the name of that antibody. And this has a Fab from this antibody, which is called PG16. And this is really remarkable, I think, because it's going up to 100%, and it's amazingly potent compared to these antibodies. Very close relatives of both of these antibodies are in clinical trials in humans, and this is how much better we could do. Over 100-fold lower in concentrations. That means less side effects. It means making it practical. And I think it means more than that, and I'll try to show you that on this slide. So what have we shown, and what's next? I think from the basic research point of view, this virus is an amazing machine that has evolved so well. And I think that what we showed was there is a significance to HIV's low spike density. It's a strategy to evade antibody-based immune responses for the reasons that I showed you. And I don't think that was necessarily obvious. We thought that that might be the case from looking at things and thinking about it, but we needed to do an experiment to actually suggest that that's true. And so I think this is really interesting from a fundamental point of view. I think it shows us that the ideal anti-HIV therapeutic for passive delivery would utilize avidity, that just means binding twice, within a single spike, so it would achieve intra-spike crosslinking, which, remember what I mean by that, it would bind with both arms to a single spike. That would reduce the concentration you'd need for sterilizing immunity to get rid of this virus, and it would make HIV's low spike density totally irrelevant. So that weapon that it has would have to go away. And then I think that this is something that's very exciting that we want to explore in animal models, which is, I think this is analogous to the idea of using several drugs during this anti-retroviral therapy, which is that HIV can't mutate fast enough to counteract several drugs. If you had simultaneous binding to different epitopes, meaning one of these Hetero-diFabs or even a trivalent heterofab, that would reduce the sensitivity to these mutations in the envelope trimer. I think it might even abrogate them. I think maybe it's possible that HIV could not mutate against something that was binding to several different places at really much lower concentrations than would normally be the case for an antibody, because these things are 100-fold, sometimes 1,000-fold more potent than their parental antibodies. So in the next experiments, we're going to replace the double-stranded DNA in the best Hetero-diFabs with a protein linker. I told you that proteins aren't a great linker, but there are proteins that are relatively stiff, like DNA, and we've found one here that we're doing some experiments with now, and it looks very promising. The reason we'd like to replace it with protein is that that would allow us to manufacture it more easily, but also it would allow gene therapy methods to deliver the protein as encoded by a gene. And so then we're going to evaluate the efficacy of these reagents by putting these proteins into animal models, and the animal models will either prevent HIV infection or actually treat an ongoing infection. And the ultimate goal is clinical trials in humans. And so with that, I want to end by just emphasizing again that I haven't talked about it so much, but this all started from the desire to understand this amazing machine, which is HIV. Unfortunately, it's a terrible bane to human existence, but it is so highly evolved that we can learn all kinds of fundamental things about it that are really fascinating to biology. And then also, from that leads translational applications. And without the first, the fundamental biological research, there wouldn't be any translational applications. I really believe that. And so I want to say that the arms race and everything we're doing in the lab is possible only through funding from the Gates Foundation, Howard Hughes Medical Institute, the NIH, and then these facilities that I mentioned earlier that we have at Caltech through places like the Gordon and Betty Moore Foundation, Agron Institute, and the Beckman Institute. And then I want to thank my lab and acknowledge them. I've tried to put people's names on the slides who did this work, but I haven't really emphasized it because really this is an enormous team effort. I have an amazing lab which has members of the professional staff, someone doing electron microscopy. We have postdocs and graduate students who work with undergraduates and high school students. These people are helped by an amazing set of technicians. None of this would have been possible without everyone working together, and also a lot of things done in a separate laboratory at the Protein Expression Center in the Beckman Institute. I have a wonderful collaborator at Rockefeller University, and then I mentioned the work. I'm only mentioning a few previous lab members because these people did some of the work here. The work I talked about was mostly done by Rachel, about the intra-spike crosslinking was mostly done by Rachel Galamidi, but I want to emphasize again that this is really a team effort. I'm so sorry I wasn't able to tell you about what other people are doing. I think they're equally exciting. And so if you see any of these people in the audience, please come up and thank them for these are their names. And so I'd like to close by leaving this slide up there so that in case you see one of them, you can tell them what great work they're doing. Thank you.